I developed an interest in understanding the brain already before actually high school. I found this particularly interesting just reading about particular disorders like neurological disorders, disorders with behavioral aberrations, injuries that cause disorders. That's just makes you realize that we define ourselves by the way we are thinking. Our brain is so important in giving us a personality that we take for granted. But this is a fragile situation. Aberrations, disruptions, injuries, they can tilt that balance. And suddenly we, in the worst case, lose ourselves. So that basic question, what makes us human? How does the brain define who we are? How do aberrations change that? That is what brought me into neuroscience. In order to understand synapse development, we of course have to use special use to analyze synapses. And these have been, I would say, the real start of the field was imaging synapses. And that's already now 70 years ago with the first electron microscopy. Because a lot of biology depends on first actually visualizing what you're looking at. And we still, to this day, use electron microscopy to a large extent to actually see synapses at the highest resolution that's available to us. But in order to get molecular insight, which is the core interest of my lab, we have to use molecular tools. In our case, this includes protein biochemistry, so we purify synaptic components and purify the protein complexes. And that's one way how we get at the molecules. Other ways that are important for us to analyze and understand synapses is actually to look at them as they develop. And you can do this. You can take neurons and you can visualize live neurons that are in front of you in a microscope and image them. And so that allows us to analyze the structure and function of proteins that contribute to this process and allows us to visualize this process. And I think another key component for us that we need to understand synapses is to move not to only analyze synapse structure and formation, but also synaptic function. And synapses function because they allow electrical transmission in the brain. So we have to be able to analyze that. And you can do this using electrophysiology. That's a big word, but ultimately all it means is that you poke electrodes into cells and do recordings of electrical stimulations. And then you can get a precise pattern of electrical activity and how synaptic transmission occurs. Again, essentially online, live in front of you as you're doing these recordings. And so we have multiple tools from protein biochemistry to live imaging to electrophysiology to look at synapse assembly and synaptic function. Ultimately, the goal, of course, is to understand what goes wrong, what's happening on the molecular level that triggers dysfunctional synapses. We need that information because we need to find where we can intervene, where there are molecular points where we can actually have a target that can, for example, be altered by a drug, I'd say, enzymatic activities that we may be able to change by providing medication.